Hello everyone. So today I am going to discuss the topic spermatogenesis. So before going into the description, let us just know what does this term mean. So genesis, we know that this term means formation or generation, and the spermato, that means the sperm formation. That is, today we're going to learn the entire process of formation of sperm cells. So we all know that sperm cells are being produced in the testes of male. That is, the sperm cells are the male gametes. So spermatogenesis, this process takes place within the seminiferous tubules of the testes. So here is the picture of the sperm cell. So the sperm cell consists of a head, a middle piece, and a long tail. So we are going to know that how this structure is being produced with the different components. So the spermatogenesis process includes two main stages. Number A, formation of the spermatids, and number B, spermiogenesis, or the transformation of the spermatids into spermatozoa. So this is basically a picture showing an egg and the sperms that are going to fertilize this egg cell. So now first of all, formation of the spermatids. First, number one, it is again divided into three stages. Number one is the multiplication phase, number two is the growth phase, and number three is the maturation phase. Number one, multiplication phase. So by the term itself we can understand here the cells will multiply. Okay, so in this phase the primary germ cells divide repeatedly by mitotic cell division. Now we know the mitotic cell division, mitosis. By mitosis, the parent cell divides into two daughter cells. And the most important thing to remember is that in mitosis, the number of chromosomes remains the same in the daughter cells as that of the parent cell. So in the multiplication phases, the primary germ cells divide repeatedly by mitotic division and they produce a number of cells which ultimately becomes the sperm mother cell or spermatogonia. So the spermatogonia or the sperm mother cell, it consists of twice a number of chromosomes. Next phase is the growth phase. So in this phase, the growth or the cells attain the maturity. So in this phase, the spermatogonia, what happens to it? It increases in size. Now how? By accumulating the nutrient materials. So now the question comes to your mind that where from this nutrient material comes up? So this comes up from the Sertoli cells. So these are the components present in the testes. And if we study the histological sections, we will find that within the seminiferous tubules, we will find the Sertoli cells. So these are now known as, after attaining the growth, these are now known as the primary spermatocytes. So these are also of twice a number of chromosomes. Last one is the maturation phase. So the primary spermatocytes, now what happens to them? They divide by meiotic cell division. So here we should remember that the primary spermatocytes, they undergo meiotic cell division. That means from one primary spermatocytes, 4 daughter cells will come up having n number of chromosomes. That means the chromosome number in case of primary spermatocytes, it is twice n. However, in case of the secondary spermatocytes, that is the daughter cells of the primary spermatocytes, it will get reduced to the half. So here the primary spermatocytes will produce the secondary spermatocytes having the n number of chromosomes. And both the secondary spermatocytes will again undergo the second meiotic division. We know that the meiotic cell division consists of two phases. First meiotic division and the second meiotic division. So in the second meiotic division, however, the chromosome numbers of the daughter cells will remain the same. That means the spermatids will have n number of chromosomes. Next phase is spermiogenesis. So the process by which the rounded or the non-motile spermatids get converted into the motile sperm cell is called spermiogenesis. So during this process, actually what happens, we know, always we have seen that cells are a round or oval shaped structure, but if we see into these sperm cells, these are quite elongated structures. So how this happens? This is because of the process of spermiogenesis. So here basically nothing, not any cell divides, but here what happens, the modifications takes place in the spermatids. So what type of modifications? Let us just see. Number one is the nucleus will shrink by losing its entire fluid content, and the chromosome, that is why they will come very and they will closely packed with each other. Next what happens, the mitochondria of the spermatids, they also accumulate and they get spirally twisted. So if we look into this, see here, so this is the head portion, and here where you can see the nucleus, and here you will see the mid, in the mid piece, this is the mitochondria, which is highly twisted in the mid piece. The Golgi apparatus. So we know that a living cell, it consists of a nucleus, mitochondria, Golgi apparatus, endoplasmic reticulum, right? So many cell organelles are present. So here the Golgi apparatus, specifically in the sperm cell, it gets modified into a cap-like structure, the chromosome. So here in the picture we can see that this is the, here in the head region, at the tip of the head region, we can see a chromosomal vesicle. So this is the acrosome. Okay. So this is the modification of the Golgi apparatus. So this is the acrosome. Now, what is the function of the acrosome? So this is basically, it helps during the fertilization process, and we will see it in details in the fertilization topic. Next, most of the cytoplasm of the spermatids are also eliminated. So in this way, the sperm attains the structure, elongated structure. So here you see, these are the two pictures taken under a light microscope. Here you can see these are the sperm cells, and these are stained by the Kumesi brilliant blue stain. Okay, so here you can see a bluish tinge is there at the outermost layer. So this is the acrosome. If the acrosome is present, then you can see this type of bluish color layered towards the outside of the head. So here you can see there is no such layer or darkly colored layer is present. So this is without the acrosome. So whatever we have discussed, it is just elaborated in this schematic representation. So first of all, we have the primary germ cell present in all organisms in male. Okay, so this is twice n in number, and it will then divide through mitotic cell division, and it will lead to the formation of spermatogonium or the sperm mother cell. Now this will again divide through mitotic cell division, and it will lead to the formation of primary spermatocytes. After this, the primary spermatocytes will undergo division. Which type of division? Meiotic cell division. Okay, so through meiotic cell division, it will lead to the formation of the secondary spermatocytes. So now we can see here that primary spermatocytes is twice a number of chromosomes, but when it comes to the secondary spermatocytes, it gets reduced to the n number of chromosomes. Then the secondary spermatocytes will again undergo the second meiotic division, and it will lead to the formation of 4 daughter cells. These are the spermatids. So there is a huge difference between the spermatid and the sperm cells. Please note it down. that the spermatids are having oval shaped structure with endoplasmic reticulum, Golgi apparatus and everything. But in the sperm cell, in the matured sperm cell, the shape becomes elongated and the Golgi apparatus it get modified into the acrosome. So finally we get the sperm cells through the modification of the spermatid, and the process is known as spermiogenesis. So this is the entire schematic representation of the spermatogenesis. So this is a sperm cell. Okay, this is taken under a light microscope. So the structure of the spermatids and its maturation to sperm cells. So nearly about 35 days is required for a sperm cell to get entirely matured. So first of all, you can see in the first week, this is the spermatid with the nucleus, mitochondria and Golgi apparatus. So with time, it gets modified little, and finally in the fifth week, that is after 35 days, it takes the shape of an elongated cell with a specific head, neck or midpiece and the tail region. So thank you everyone for your kind patience.